Okay, we are back again, covering Chapter 9. This is the second part, variation of Mendel's laws. So we're going to cover some more things that are going to be on the test for us. And go from there. So we talked about Mendel and his P's and how to do what some people call box babies and probability of inheritance, independent assortment, random assortment, things like that in the last lecture for this chapter. So now we're going to cover things like incomplete dominance and why we see certain variations from Mendel's laws. So in the P crosses, you would always get one or the other, and that's a complete dominance. But for some characters, there's incomplete dominance, which kind of the hybrids are in between the two parental varieties. So expression of both alleles occurs. And so red flower, white flower makes pink flowers, but then when you make two pink flowers, you get red, pink, and white. You get all three phenotypes. So this is a type of incomplete dominance where both are kind of expressed, and you get kind of that blending look. So it's not surprising that they came up with that blending hypothesis. But, of course, that blending hypothesis wasn't supported because the F1 generation would produce all three in the next one. But an example your book gives is high levels of cholesterol occur in the blood, and heterozygotes have intermediately high cholesterol levels. So here we go, homozygous, right? And so you get a normal cell, and then you're heterozygous, you get a mild disease, and homozygous recessive does not make any receptors. So you have that cholesterol. And so we can only carry two different alleles for a particular gene. But that's not, you sometimes get more than two alleles in a population. Right? So human ABO blood groups, phenotypes involve a single allele for a single gene. So the human blood groups are A, AB, B, and O. Okay, so A and B are both expressed at the same time, and that is known as codominance. So go back to that flower, and so you have a red flower and a white flower, and if they were codominant, it wouldn't necessarily end up pink, but one cell would be red and the other cell would be white, and so it may look pink or it could be blotchy, all sorts of different things, but you would get both alleles expressed. And so in our blood groups, we have more than one allele because we have that O allele. And so in codominance, right, it just shows that both phenotypes are expressed at the same time instead of some sort of blending. And we often get with blood, you should know your blood type. You've probably heard A positive, A negative, B positive, B negative. Okay, and that's an antigen that occurs on blood and how well it clumps, and that's why you need to be compatible with the same type of blood before you donate blood, because otherwise your body will see the other blood as an infestation and try to get rid of it. So that's why they need blood donors. It has to be the same type of blood. So pleiotropy is another problem that causes variation with Mendel's laws. And so you can have one gene and it makes lots of different characteristics. So we have sickle cell as a single allele, and it affects hemoglobin in the blood cells. I'll show you a picture here and why they call it the sickle cell. And so you have normal blood cells, and then you have some sickle cells. And you see this very often in Africa. One allele will produce these sickle cells, and the sickle cells don't carry oxygen nearly as well as the regular blood cells. So if you're homozygous recessive for sickle cell, you have a very hard time moving or doing anything because of the lack of oxygen. If you're homozygous dominant for the normal condition, you can carry oxygen normally. If you're heterozygous, then then you can still carry oxygen, but just not nearly as efficiently. I believe there's a football player that every time he goes to Denver, Mile High Stadium, he has to sit on the sidelines because he has sickle cell, and he can't. He could die if he played without that type of oxygen. Now, interestingly enough, in Africa, the heterozygote condition is the most dominant, and that is because sickle cell helps ward off malaria, which means it makes it more difficult for malaria to get into your blood. So someone who's homozygous dominant for normal cells contracts malaria very easily. Someone who's homozygous recessive doesn't contract it at all, but they can't move or reproduce. But a heterozygote has a good resistance to it, and they can still move. So in that case, it becomes the dominant genotype. So different things from sickle cells, kidney failure, heart failure, spleen damage, all sorts of things can happen because of sickle cell. So when you have a lot of characteristics, and that results from polygenic inheritance, which is a single phenotypic character and results from the additive effects of 2 or more genes. So now you have multiple genes creating a single character instead of pleiotropy, which is one gene that creates several characters. So human skin color is polygenic inheritance. Okay, so you get this kind of mixture and you start to see the mean around a certain pigmentation. Now, every phenotypic expression is the product of a genotype times the environment. So the environment has a big effect on what we can look like as well as our genotype. So skin color is affected by exposure to sunlight. Cancer can be hereditary with certain environmental components. And then identical twins can show differences, and all sorts of different things where the environment can cause a different phenotypic expression. And there's your skin color, right? So that is part 2, and we're going to, part 3 is going to go over the chromosomal basis of inheritance. So I know part 1 was long, but these next ones are going to be kind of short.